Hello, my name is Sarah King, and my poster is over gene expression changes in mesenchymal stem cells due to different pathologies, and I specifically zoomed in on the FLRT3 gene. I looked at this project because mesenchymal stem cells are currently under clinical exploration as a therapeutic approach for people who have tissue damage or need regeneration. They'll just inject the stem cells in that area, and they've shown to be very effective, and they have tons of potential for the future. But one of the problems is their effectiveness remains limited due to the fact that in some people it's causing tumors and even more inflammation and fibrosis after the cells have been transplanted in the body. And basically the aim of this study was to figure out why this was happening in certain people, specifically people with diseases, and understand why their body is rejecting the stem cells when other people's isn't, and to see if we can find a common gene that could potentially be causing this problem, and maybe understand it on a deeper level enough to have a solution eventually, so that people that have pre-existing conditions can still receive stem cells and notice potential benefit from them. So to start out for this project, I found 3 different sets of data with different diseases that were comparing control DNA, so from a healthy person, with disease DNA. And the 3 diseases that I looked at were osteoporosis, acute myeloid leukemia, and lung cancer. And the data was specifically looking at the gene expression of the mesenchymal stem cells. So I took the data and I ran it through the TAC software, which will give me a gene level analysis of the upregulation and downregulation of the data. So I exported the data to an Excel file. I changed the data to linear fold changes so it'd be easier to read, and I organized the fold changes from greatest to least, and then from least to greatest. So when I was looking at the largest fold change, I was seeing what genes were upregulated in disease patients compared to the control. And whenever I was looking at the smallest fold changes, I was looking at what genes were the most downregulated from the control to the diseased patient. So I wanted to get 2 perspectives just to kind of get that well-rounded view. So I organized them in Excel, and I was able to find some genes that were similar in all 3 diseases. And I wanted to organize my poster kind of looking at the data from a large scale and zooming into a smaller, very gene-specific scale. So on the far left here you see a PCA graft, which is just demonstrating the overall gene expression differences between the healthy mesenchymal stem cells and the diseased. And the blue represents the control, the red represents the diseased. You can see each of the 3 diseases there, and just kind of the differences and how the data is already showing that it is definitely changing in a control patient versus a disease patient. If the dots had been clustered together, there would have been similarity, but luckily there was differences. So I dug deeper and I looked at genes at a level on their specific name through the Excel spreadsheet, because the TAC software allows you to see genes on a name basis. And I organized them here in this chart on their fold changes, and the numbers represent the rank of what the fold change was. The top box on the chart represents the top genes that were the most upregulated. So the higher the rank, so for example rank 11 with the PNMAL1 gene, shows that this gene was ranked 11 on the highest upregulated genes for the osteoporosis dataset. So that's what these numbers are representing. The bottom 2 charts are showing the most downregulated genes. So for example, the ranks 1, 2, 5 that you see for acute myeloid leukemia, those were the top downregulated genes for that disease. So the first box is upregulated, the bottom two are downregulated. I was able to find more downregulated common genes for the 3 diseases than upregulated, but I thought it would still be good information to have both on my chart. I also put the data in a plot, a graph format, so you can almost visually see the differences in the significant genes in figure 3. So that's more of a visual representation of what is going on. And at the top, I just wanted to show the specific gene of FLRT3. This is kind of the one that I zoomed in on, and you see the purple dot. This gene stood out to me because of the fact that it had very high results, specifically a high rank for lung cancer, a high rank for AML, and a high rank for osteoporosis. Compared to the others, it had more of a higher upregulation, and this gene had a very specific function that caught my attention, so this is why I chose to specifically center in on this one. It's responsible for cell-to-cell adhesion and receptor signaling. So when I look at these mesenchymal stem cells and how they're going wrong in people with diseases, it's because they're causing tumors and fibrosis, and both of these irregularities are caused from increased cell-to-cell adhesion, receptor signaling. That's kind of the pathophysiology of a tumor, is uncontrolled spreading and growing, and obviously that involves increased cell-to-cell adhesion. So I thought it was very interesting to find a specific gene that was highly upregulated and had an interesting function that seemed to match how this would be causing tumors. So I highlighted this in the scatter plot above, and I just showed how this gene was upregulated. You can see it's kind of separated from the rest of the cluster on the top of the graph. So that's what those are highlighting. You can also just kind of see in general which diseases had a lot of genes that were upregulated, and the significant ones are in color. So speaking of function of genes and what's going on, I highlighted what I just talked about in the chart on the far right. I talk about the FLRT3 gene, which is the one that I really felt like had the most potential to pursue further research with. So this is kind of the one that I really wanted to base my project off of, but in the process of getting to this decision, I also looked at 3 other genes that also had interesting functions that could be potentially related to the tumors that are being formed in people with disease. So I found the NPR3 gene, the PNMAL1 gene, and the CTNNB1 gene to also have potential functions that could be related to why these cells are developing tumors. So I kind of talked about that there in the chart. That's probably the basis of this whole poster, was understanding how these genes are changing and the function of these genes, because when you understand those 2 pieces, you can understand why a treatment plan is not working for someone at the genetic level. I had a very fun time doing this project. I think if I were to expand it further, I would definitely look at other diseases and include them in this project to understand, okay, are these genes also similarly upregulated or downregulated with other pathologies, or is this just a common finding for these specific 3? I think adding more and more diseases will increase the validity of my data, and maybe even highlight some more genes that are similar in all of them. So if I were to take this project further, this is kind of the route that I would be considering. I would also like to do a quick shout out to Dr. Y for all of his help in this project. I really appreciate it. If there's any questions, feel free to reach out, but this is my poster, and thank you so much for listening. Have a great day.